Hi friends, so now I'm back with my next video, and in this video we are going to talk about DNA cloning. So what is DNA cloning? It is the process of making multiple copies of DNA, multiple copies which are identical to each other. So we are going to talk about the technique, the different steps that you need to follow to do this cloning, and how do you select the plasmids which contain your DNA or not. So please make sure to like, share and subscribe my channel. So what is DNA cloning? So as we have talked about, it's the process of making multiple copies which are completely identical to each other of your gene of interest, of your DNA which is of your interest, of a particular piece. And it can be done by various methods. So two of the most common method is, first is the cell based technique, in which you take your plasmid of DNA and then you put your gene, and then you amplify it using using a host cell system like the bacteria, or like different there are yeast system which are also used to grow this. So you use different host system to grow your DNA. And the another one is using the polymerase chain in reaction, in which you put your primers, you put the enzymes whatever you need like the polymerase, and then you use different temperature cycles like the high temperature for denaturation, elongation and extension for the amplification of the DNA by the machine, like the machine technique. So these two methods are the most common approaches which are used to do the DNA cloning. And for the cell based technique, you need a vector or a plasmid to carry your gene of interest into the host cell, because you cannot just add your DNA into the host system, because then it will be degraded. You need some kind of a vector that can replicate your DNA and also prevent it from degradation. So plasmids are good to do this because they are very stable in replication. So, DNA cloning, why we need to do this? Because this is the most important stage, the first stage of for most of the genetic engineering. Like, suppose you are going to screen hundreds of genes, and you want to know the function of one of your gene, and you need to make the DNA libraries. So to make the DNA libraries, you need to do the DNA cloning. And for the PCRs, where you amplify your DNA, sometimes you need to clone your DNA, gene of interest, into a vector, and then you isolate your plasmid DNA from the host system, and then you do the PCR amplification of your gene of interest or of your DNA. And then for DNA sequencing, also you need to clone your DNA to replicate it, and then you can send it for sequencing. So, like, you need to make your construct for the sequencing, for the DNA libraries, and for the PCRs. So DNA cloning is the first stage of everything, and it's very important to know the concept and how can you do it. So now we are going to talk about the process of the cloning. So first process is, as I have talked about, you need to insert your target DNA in a vector. So here you can see a cloning vector and a target DNA. So how can you insert your target DNA into this cloning vector? So this can be done with the restriction enzyme. So what you do is you cut your DNA with some restriction enzyme. Suppose I am talking about two restriction enzyme like EcoR1 or BAMH1. So you need to cut it in a way that they are very close to each other. So generally in the plasmid you have a cassette which contains different restriction sites. So you need to select the restriction sites from that place, so that it does not remove anything from the plasmid vector, and you can insert your target DNA in the same place. So suppose you cut with the two restriction enzyme and it leaves overhanging bases. Then you need to cut your target DNA with the same restriction enzyme, which which will cut at the same restriction enzyme, like leaves the same overhanging ends. But because contains the same overhanging ends, you can ligate them, because then they will form the base pairs with each other, because A will be opposite of T, because there are same overhanging ends. And then you can ligate them using ligases. And in the same process you can insert the insert your bases, whatever you need to do to create the restriction enzyme specifically, or the whatever, like the site you want to create, you want to add the tag, you can use all these primers to generate this. And then once you do that, there will be some which contains your target DNA and the other which does not contain your target DNA. So now you have this your plasmid, your vector, and the next step is to replicate them, which you can do using a bacterial host system. So what you need to do is to transform these plasmids into the bacteria. So this is done by a technique called transformation. So for this, what you need is a bacteria which is competent enough to take up your plasmid. So the normal bacteria, they cannot take up your plasmid easily. You need to make them competent by the calcium chloride or by electroporating techniques to make them competent to take up the plasmid. And once they are competent enough to take up the plasmid, you can put this plasmid using different methods. So first is the heat shock method, in which you give the cycles of heat and cold to make the membrane porous so that your plasmid DNA can go inside it. Or what you can do is you can use electric fields to make the membrane porous, and then it takes the DNA inside. The process is called electroporation. So whether you do it by the heat shock method or by the electroporation method, you are like transforming your recombinant DNA into the bacterial system. So there will be different type of bacteria. The one which does not contain anything, like which did not took up the plasmid, the one which took up the plasmid, but one contained the recombinant one and other the non-recombinant one. So now the next step is to find the transform cells. So what we do is we will remove this one, but we will take this one. So how we can do it? By the action of the antibiotic selection. So the vector that we have contains a ampicillin, like antibiotic resistance gene, right? So suppose in this case we are talking about the ampicillin. So because the plasmid contains the ampicillin resistance gene, suppose, so the bacteria which does not contain the plasmid will be the sensitive to the antibiotic, and then they cannot grow on the plate which contains antibiotic, right? Because if it contains antibiotic, the bacteria will die. But only the bacterial which contain the resistance to that antibiotic will grow, and that comes from the plasmid DNA. So only the transformed one which will be appear on the plate. So then you can grow them, and then you need to select the one which is your gene of interest or the one which is the non-recombinant one. So this you can do it using restriction enzyme, because by inserting this you can know when you cut them with restriction enzyme which size of band you will get, which size of bands will appear, whether there will be 2 bands, 3 bands, 1 band. So based on that you can confirm your results, which is the recombinant one and the one which is not recombinant. And once you know which the recombinant one, you can amplify them, you can grow the bacterial stalks, and then from the bacterial stalks you can isolate plasmid DNA using alkaline lysis method, or there are some kits available to do the isolation of plasmid DNA from the bacterial stalk. And once you have that bacterial stalks, the plasmid DNA, you can amplify that plasmid DNA using the PCR, and then you can also cut your gene of interest from the gel by using the restriction enzymes. And then you can, with that gene of interest, you will have multiple copies, and then you can make DNA libraries or the other things that are required for the genetic engineering technique. So I hope it's clear, and now I will talk about why we are using plasmids as the cloning vector, because there are other systems as well that can be used for the vectors, but the plasmids are good one because first thing is they are in small size, right? You can easily manipulate them using the restriction enzyme, using the primers. You can add some bases, you can remove some enzyme restriction sites and all these things. So they are easy to manipulate because they are in small size. And as I have talked about, they are circular in size, so they are much stable, because the linear ones, they can be sometime degraded, and they can be recognized by the host system and they can be degraded. But because they are circular, they are more stable, and then they replicate independent of the host cell. As you have seen before, they are the transform bacteria, they contain the plasmid DNA as well as the bacterial chromosomes separately, bacterial DNA. So they can replicate independently of the host cells, they don't need to be recombined into the host cell machinery. And because, as I have told you, they replicate so fast, they are in small size, so you can have many copies of the plasmid DNA, like small size copy, like some plasmids are low copy number, some are large copy number, which means that they can replicate and make multiple copies of the same DNA. And because, as we talked about antibiotic resistance, that they contain the antibiotic resistance gene, which is easy for them, for us to detect the transform cells or the between the non-transform cells, like the one which contain the plasmid DNA and the one which does not contain any plasmid DNA. So I hope it's clear. You now understood how to clone your piece of DNA into the plasmid and then grow them, make multiple copies of it, and why plasmids are good vector. And if you have any doubts, please let me know in the comment section. And please make sure to like, share, and subscribe my channel. Thank you.